I'm going to talk about coarse graining. Coarse graining is a very, very important aspect of multiscale theory and simulation. I think if you look in the literature these days, take a journal like Journal of Physical Chemistry, Journal of Chemical Theory and Computation, Biophysical Journal, you'll see increasingly large number of papers that are using these coarse graining ideas. And in a nutshell, it seems very simple what coarse graining means, but actually at a deeper level, if you try to affix some real meaning to it, meaning something tied to real theory, it's not so simple. And that's I'm going to spend part of the talk today talking about that, and essentially what we have done, although I will try to tie in ideas from other groups around the world. I certainly don't have the time to do that. And then I'm going to finish talking about some of the applications of the coarse graining, and actually two of them you'll be playing around in your computer lab, so that's why I wanted to sort of introduce those to you. So what coarse graining is, sort of at the initial pass, is, you know, we take an all-atom system, something where you've resolved every atom of the system. Maybe this is, unfortunately, hey Andrew, will you get me a new laser pointer, please? This one's dead.
Oh, it is?
Yeah. Maybe batteries, I don't know. So if you resolve all the atoms, this is just a simple helical peptide. We map some number of those atoms in the amino acids into these funny-looking coarse-grained things we call beads. And beads, presumably just because it kind of looks like a necklace. Okay, but I call them coarse-grained sites or coarse-grained beads. These are different examples. This is a lipid bilayer. You can see the lipid. We didn't really show bondedness here. I think this is a solution of sugars, of carbohydrates. This one we'll come back to later. This is an actin filament, or a piece of an actin filament. So there's these are whole proteins here, and you can see they've been very, very aggressively coarse-grained into simple objects. Okay, so I want to spend time trying to not do too much math, but talk to you about how we understand this in a somewhat deeper way, or actually I should say a lot deeper way. And the idea is that you want to somehow adhere to some sort of a theoretical foundation. And for most of what we do in molecular simulation, that theoretical foundation is statistical mechanics. So statistical mechanics tells us that we would like to evaluate in some way this Boltzmann distribution by averaging over the atomistic degrees of freedom. So R here is a very large vector of particle coordinates. Okay, it's not one, it's many. You'll notice that this is 1 over kT, the temperature. And statistical mechanics tells you that if you could just do that calculation, there's a normalization factor, which I'm not going to carry around. It's for the ideal gas. So we're not going to worry about that right now. If you could do that calculation, it ties you to the free energy. That's what F is, the Helmholtz free energy. And generally, we don't do that calculation directly when we do molecular simulations. We calculate some other thing like an average of structures or a heat capacity or something related to calculating properties. But in that equation there really is the heart of what we want to do. And the problem is, and maybe many of you have been confronted by this problem, is that this, for a non-trivial system, say a biological biomolecular system, that vector R is a huge degrees of coordinate. Do you have the laser pointer?
Right there.
Oh, okay, thank you. It is a huge number of coordinates, right? And so this is a huge integral, and the sort of perversity of statistical mechanics, if you will, is, you know, you're sampling many of these configurations. Most of them are irrelevant. Most of them don't matter because this exponential factor that depends on the potential energy function kills most of those configurations. Because you see there's the potential energy, so if the potential energy gets high in some sort of a region where you know, you don't want to be, then this kills it, okay? And then on top of that, for you know, sort of non-trivial systems, this function itself is a very complex function. So a protein you know, force field, like from charm, this is itself sort of exponentially complex. It's got many minima, and And so, you know, things like the protein folding problem, we try to figure out exactly, even if we can find the minimum energy of this, it would be a big challenge. Okay, so what I mean by statistical consistency is when we do coarse-grain mapping, okay, and this is just a simple example of a lipid molecule. It's been used many, many times in the literature by us and others, and you can kind of see at this level of coarse graining, maybe you take sort of 3 carbons, map them into one of these, and of course I'm not really showing the hydrogens that go along with it, but they come along too. You can see the head groups. So we're coarsening. We're reducing the resolution of the problem. What we would like to do when we do this is to maintain this connection to the real statistical mechanics. In other words, we would, in our opinion, we think it's very important to not depart from this connection to real theory. We have avoided largely the sort of the impetus to just create a modeling. In other words, just a set of objects like this that have some properties. We're trying to actually connect the real molecular interactions to these coarsened configurations. The important part here is this number of variables is a lot less than the atomistic one, okay? I mean, in this case, it's not hugely less, but it can be a lot less. So what that means is that we need to think about how to do this transformation. We need to think about how to do it rigorously. And in science, in theory, theoretical science, generally the rigorous route won't lead you anywhere. I mean, it leads you somewhere conceptually, but in a practical sense, you won't get anywhere. It's just a frontal assault. So we're going to have to figure out a way to get around that. But we're confronted with 2 main questions. The first, how best to do these mappings. And so Martin McCullough, who's going to come after me, is going to talk about this problem of these mappings to coarse-grained objects when you're really looking at big proteins and you want to very aggressively coarse-grain them. Here, you know, that kind of made sense, this mapping, but I could argue, you know, how do I know that say maybe it shouldn't have been 2 to 1 or 4 to 1, right? So how do I know how best to do this mapping? And then the second, yeah, sure.
So when you say mapping, like what are those that you're actually mapping?
I'm going to show you in a minute the actual mathematical form of the mapping. Yeah. Okay, so, but I'll give you the answer ahead of time. For example, this would be the center of mass of these carbons, the center of mass function. Yeah. So, so the second part is once you have the mapping, what is this effective interaction? Because it's no longer the atomistic interaction, right? It can't be. It's been something that's been reduced. You'll see really what it is now is a very complex function in principle that's more of a free energy surface. And the reason for that is that when you do this mapping, you're effectively integrating some of these variables out of the problem. You're doing integrals over some number of these, and that integrates over their motion, their fluctuations at temperature, and folds their properties into this function. So that's a very difficult function to know. Okay, so let me show you how you do this as a frontal assault, okay? And so let's forget about the mapping part for a minute. Let's assume you feel pretty good about doing that, you know how to do it, all right? And that's not always true, but let's assume that you do. So the question is, how formally should you determine this thing? Well, let's go back to the beginning, okay? This is our statistical mechanics expression. I should point out something, though. Already, we're making an assumption that we're at equilibrium. This is an expression from equilibrium statistical mechanics. Very few people, and if I have time, I'll talk about this. Very few people have thought about non-equilibrium problems in this sort of thing. Okay, here's your question. Now, this is a little bit meaty, but let me show you. The way we introduce collective variables, it's kind of trivial in some senses. We have mapping functions that take the atomistic coordinates and map them onto some point in space, and every time that mapping equals where the coarse-grained coordinate is, and this is really a product of many of those for all of the coarse-grained variables. I'm just writing it shorthand, right? So understand this as many delta functions. You see, I can insert this into that formula trivially, because if I integrate over everything, it's just equal to 1. Integrating over a delta function is equal to 1. And so to answer the question more concretely, every one of those m functions in this case would be the center of mass of the instantaneous center of mass of those atoms that go into that coarse-grained site. Okay, and we know that center of mass is a transformation. You probably learned this in high school physics or something. The center of mass of an object. especially an object of point particles, is pretty straightforward. It's a linear transformation. So now the trick we do is we switch the integration order. My mathematician friend Jonathan probably cringes when we do this too cavalierly. You know, mathematicians really don't like it when you switch order without thinking about it, but we always do it and never got away with it. We always got away with it somehow. So, but here's the part that's important. Now we want to impose this thermodynamic consistency. We don't want to lose the connection to the real statistical mechanics. I have to warn you, this equation, there's normalization factors for the ideal gas where the interactions are turned off that we kind of fold into these definitions. I just don't like to carry around a lot of constants. And if I do that, then I see what the formal definition of this coarse-grained interaction is. It's a little bit complicated, but let me explain it to you in words. It actually is harder, okay, harder than the straightforward problem in some ways, okay? So remember, the straightforward, meaning straight frontal attack of just doing a molecular simulation, okay? We would sample from molecular dynamics, Monte Carlo, something. We would keep calculating the trajectory and averaging over this distribution. Now, for this coarse graining, I'm doing that. See, I'm integrating over all the atomistic degrees of freedom. But on top of that, this delta function in here tells me I only register the data every time this mapping function puts the atoms at some place equal to where the coarse-grained variable is supposed to be. Okay, so this is like a filter function, right? I'm doing lots and lots and lots of atomistic calculations. This filters out and only allows the atomistic ones to project onto the coarse-grained variables for certain places. Okay, so this is a very difficult problem. You would not be able to solve this from a frontal assault, all right? And I'm going to show you how we have done it, but I'll sort of talk about other ways that people try to get at this. The most important thing, though, that I want you to realize is now this is the definition of what we call a potential of mean force. Okay, sometimes we abbreviate that as PMF, potential of mean force. It's a little bit different than the potential of mean force you encounter in sort of the standard literature because it involves a lot of coordinates. The typical potential of mean force is, let's say I take two particles in a condensed phase system and I decide I want to calculate the effective interaction between them averaging over everything else in the system, all right? So, two ions in water, okay, average over all the water fluctuations, I get this mean interaction that's left between the two ions. This is like that, except, see, there's a lot of coordinates here. So this is a multi-dimensional potential of mean force. The important aspect of this, though, is that a potential of mean force is a free energy. Okay, it's not an energy. It's a free energy. Free energy means it has entropy in it. Okay? So, and why? Because in calculating this interaction, I average over the atomistic variables. I project out where they hit the coarse-grained variables, but I'm averaging over their fluctuations. So I'm folding the entropy of the motion of these atomistic particles into this. So as far as I know, most people don't debate about this. This is the most formally correct definition of a coarse-grained interaction. All right. At this juncture, people go off in all different directions to figure out how to calculate it. All right. And I'll show you one. Some groups, so for example, the GROMACS program that you can get off the internet has a force field in it called a Martini force field. It's a coarse-grained force field. They don't attempt to do this kind of rigorous thing. What they do is fit a lot of parameters to try as best they can over a broad space of parameters to reproduce something like this. Okay, but it doesn't necessarily have any connection to any specific system. Okay, it's a fitting. Some people do what's called an inverse Boltzmann, where they try to not calculate this, but maybe just the effective interaction between two particles and find the effective interaction at the coarse-grained level that gives that. That's called inverse Boltzmann or inverse Newton. What we have done is a little bit more elaborate, but in some sense more true to this idea, is we developed a variational approach, which we call multiscale coarse-graining. And that variational approach involves, it involves a relatively simple function, which I'll explain to you. It's called a chi-squared function. And actually, Martin McCullough is going, in his lecture, is going to use a chi-squared function in a different context. But all this really is, before I explain it to you, is like a least squares fit. Least squares fit, you know, you have some points. You probably learned this in junior high. You have some points. You want to fit it by some curve, right? And so you calculate the square of the distance between the actual point and the point from some curve you're trying to fit to it over that point, okay? That's a least squares fit. See, we square. Now, what is this in our hands? Well, these are atomistic forces. This is no longer this is not the free energy, so don't confuse this from the notation before. These are atomistic forces, literally the forces that would come from molecular dynamics calculation, okay, like charm or amber, one of these things. These are the coarse-grained forces. So these are, you'll notice this is as a function of big R, so these are the coarse-grained variables. So there's been a resolution change, right? These are all atoms. This is now coarse-grained variables. The problem is we don't know what that interaction is. That's the derivative. All that is, you can prove this, is the derivative of this thing. Okay, so the forces. Or I should say minus the derivative, right? It's the forces. So the idea is you sum over every single interaction. Okay? You square it. So it's positive, right? It doesn't make sense to average a bunch of positive and negative numbers. You'll get 0. And then you average over the ensemble of, or some sort of ensemble of atomistic coordinates. So you don't have to do a complete averaging, but this is a little bit obscure. It's unclear exactly what we need here. So this methodology is a way of mapping atomistic interactions into coarse-grained forces. And it turns out that you can prove, in these papers, later you can get this presentation from us after the workshop. In these 2008 papers, we were able to prove that if you don't make any approximations to this mathematical function here, okay, and I want to talk about that because you have to make approximations, right, but assume you don't. You can prove that you, in fact, get the exact answer, all right? So the exact answer is, I should say the exact answer for the forces, for the derivatives of the coarse-grained interaction as a function of the coarse-grained particles. This is a powerful concept because it's a variational mathematics. Probably most of you have had a class in quantum mechanics. And in quantum mechanics, you learn about the variational theorem for wave functions, right? You don't know how to solve the Schrodinger equation for some really hard problem. And so you guess the solution of the wave function. And if you can adjust the parameters optimally, meaning minimize the set of parameters, you'll get as close possible as you can to the right answer. So what that means here is if I come up with increasingly more elaborate or hopefully insightful ways of representing the coarse-grained interactions, I'll get closer to the right answer. All right? And that's important because, you know, we start fiddling around with functional forms for modeling things, you know, you could get the wrong answer. You could do a lot of work and actually make the problem worse. So that's very important. Yeah?
So, might be very fast, but if I understood correctly, in this coarse-graining, we forget about the orbitals and hybridization.
Yes, that's a good question. I should have backed it. We assume that we have the Born-Oppenheimer energy surface for the nuclei. We're not assuming that we have to worry about excited states for the electrons, that we have some sort of a atomistic potential energy function that we trust. Yeah, and that's a separate question, whether we trust that or not. But we tend to hope they will get better and better and better with time. Yeah, that's it. Thanks for asking. I should have clarified that. Let me just quickly mention a couple of things that have made this algorithm work. And I think in your computer lab, you're going to play around with this. Jin is going to, he's got it working for methanol, right? Some simple system. So you're going to actually run a program that does all this mathematics. You don't have to worry about understanding all the mathematics. But it turns out that you have to now represent in these functions here, right? You have to represent them some way. Any mathematical function, you have to represent it. Okay, so we've done different approaches to that. One is it's not essential, but really simplifies the problem. Is that we assume the non-bonded forces are pairwise. Okay, so in other words, particle over here interacts with particle over here to just an interaction that depends on their distance, and then this one and this one. You can introduce more complex interactions if you want, but we assume this. And then another thing that we've done that I think has been one of the most important steps in this is we don't try to guess the mathematical form of these functions. So in this force thing, we don't say, oh, that looks like some polynomial or some exponential function like you commonly see. What we do is we record them numerically. So in the mathematics that is usually done for this are using spline functions or some sort of functions that record numerical data on a grid. So in space, pieces of distance between the particles. And all you have to do when you do that, this actually records the atomistic interactions projected onto these coarse-grained sites. These are coefficients that you have to solve for that create a continuous function. Okay, that's how these things work. And if you do that, you actually turn it into a linear least squares problem. A linear least squares problem is a very nice, it's in principle, it can always be difficult. But this is a very nice thing to have because the key word is linear. Linear means you're searching for the solution of a big parabola at the bottom. There's one unique solution. It's not some rough thing with all kinds of minima where you get stuck and you get multiple answers, all right? And finding the global minimum is extremely difficult. Linear means you find it at the bottom of a parabola. So you are certainly welcome to study this more on your own. We will always be happy to help anybody that's interested in this. It's a lot to sort of fathom. Let me give you one more sort of meaty slide, and then I'm going to talk about some applications to show you how this works. It turns out that this, and this was published first in this paper now 5 years ago, that this stuff we were doing, it was a lot deeper than we realized in terms of connecting the way particles interact with each other. And so the equations we solve, and there's no possible way you're going to understand those equations in the next 10 seconds, okay? So don't worry about that, okay? But these are the equations you solve when you solve for these interactions, okay? It turns out that if you sort of take them to the continuum limit, so you make the spacing of these grids going down just infinitesimally small, you can show that what you're actually doing with this algorithm is an inverse solution to a very famous equation if you take more advanced sort of statistical mechanics course. This is an equation called Yvonne-Born-Green equation, which what it does is it relates two-body correlations. That's the G of R function. The G of R function is the distribution. If I sit on one particle, the probability that I find another one. Okay, so that's there. This is the thing we're solving for. Okay. This now is a three-body G of R. So what that says is the statistical correlation that if I'm on this particle and there's another one and another one, so three-body correlation here, okay? And this, again, is the force. It turns out, and this is a very, very complex paper to read, but the end of it all is when we solve these equations, what we're trying to do is we're trying to find the effective coarse-grained interaction that makes this equation work, okay? And that's really important. I know it'd probably be hard for you to understand how important it is because there's so much math here. It's important because in going back to this interaction, what it says is I'm not just looking at two-particle interactions when I do this coarse-grained modeling, okay? There's a lot of methods in the literature, and especially in the polymer literature, it has worked quite well because polymers tend to be very disordered, sort of chains and stuff. And so all you're looking at is the effective two-body interaction. You can build up one of these coarse-grained potentials from that. In biological systems, three-body correlations are very, very important. What's a hydrogen bond, right? Hydrogen bond's a three-body correlation. You have something like an oxygen, a hydrogen, and nitrogen, or something like that, some sort of a hydrogen bond. So if you're building a coarse-grained model, those sorts of three-body correlations are very important. Water, three-body correlations are very important. So, okay, so let me show you, your brain can somewhat relax now. Let me show you sort of some aspects of this, and then I'll move on to other sort of results. This was one of the early applications we did. I want to show you how it works. But what I'm showing you is sort of building on before the idea of coarse-graining. This is now coarse-graining a lipid bilayer, which is more complicated. It has cholesterol in it. Okay, so cholesterol makes lipid bilayers, modulates their properties. Typically, it makes them stiffer. It changes, and of course, it's a very important, realistic molecule to have in any sort of a realistic simulation of a bilayer. I'm going to just show you sort of how the results turn out for this, and I want to illustrate something that sort of is an important point about coarse graining. So I'm going to show you results. This I showed you before, this coarse-grained model for this lipid. Water we're turning into a single particle. And I'm going to show you results for two different coarse-grained models for cholesterol. All right. This one involves 7 coarse-grained sites or beads, and that one's 4. And you can see the difference. The difference is that here, where these rings are, sort of these faces of these rings, is a little more highly resolved. Right. You can imagine that. You know, this is pretty primitive. This is really coarse-graining this thing into some strange sort of linear-looking chain of beads, but there's a lot of molecular structure in here. Okay, so the 7-site model should represent this interaction better. And so these are some results. I'll just show you. What these are radial distribution functions. So again, if you don't know what a radial distribution function is, you're sitting on a particle. It's the probability in radial space that another particle of some type is near you. All right. And the atomistic results, so for all intents and purposes, you can do these molecular simulations. The red curves are the exact results. And the blue is the coarse-grained results. And these are a subset of many possible radial distribution functions. And by the way, this is a spurious point. This has to do with a bonded interaction, which you don't worry about that. But in general, these are all really quite accurate. In other words, this is a quantitative representation of that real coarse-grained, the real bilayer at the coarse-grained level. The only exception is that this one is, if I look at that site there and look at the radial distribution function of another cholesterol molecule in this lipid bilayer with it, you'll see that in fact the atomistic model, which is the red, differs fairly substantially from the coarse-grained model. There's some sort of anomalously high peak here, and then things are shifted. And we might have predicted that, right? Because what that was using something that was too primitive, right? So the results for the 7-site model, you'll notice this shifts a little bit here, but it illustrates the point. Here we're resolving these ring faces and their excluded volume and their orientations a little bit better. But we still get away with a pretty healthy degree of coarse graining here, right? We're not anywhere near representing all of the atoms. And it comes into much, much better agreement. I mean, there's always going to be some differences. But you see here, okay. So why does this matter? Why did I want to show this to you? This illustrates, I think, the variational nature, the controllability. We made a model that we knew in our heart should be better on physical grounds, and in fact it is better, okay? Now, that's not necessarily a given if you're just doing modeling, okay? If you're just trying to dream up interactions here that might work or might not, you have no guarantee that it will be better. So this illustrates, I think, the importance of why we like to have a controllable approach. Okay, let me now, for the next 5 or 10 minutes, I want to sort of strip away some of the complexity and take a slight deviation from what I've been talking about, which has been more methodological, like how you do these calculations. I want to talk about what they tell us, all right? Using coarse graining, if you will, is sort of an interpretive tool, something that tells us, you know, what's really going on underneath those molecular structures. If you want to think of it this way, in statistical mechanics, we start with an atomistic model with a force field, okay, and we go to the thermodynamics. All right, what the coarse graining is going to do for us is give us an intermediate point along the way to kind of start taking some of the complexity out of the problem, but leave enough in there that it's still some non-trivial thing. And I'm going to show you a crazy example. And what I'm going to do is just coarse grain a fluid water. You'll do this in your computer lab, but for methanol. Methanol is a better behaved system than water. Okay, I'm going to turn it into a fluid of simple particles. Okay, so I'm turning a triatomic. There's many waters. This is just one picture here. Okay, one. I'm turning water, a liquid of water, of these triatomic molecules into a liquid of single particles, which seems crazy. But if this is going to work, that means that the physics from what real water does has to pop up in the effective interactions of these single particles. Okay, that's the spirit of coarse graining. I think this model, well, you know, it's questionable whether you really want to coarse grain water or whether you want to just get rid of water. All right, we'll talk about that, and tomorrow we're going to have a lot of lectures on what we call implicit solvent models. But let's show what this does. Don't worry about the figure on the left. Here, the red curve is the effective interaction that the single particles have in this fluid where I've taken water and mapped it into a fluid of single particles, okay? It's a crazy-looking interaction. This, by the way, is a Lennard-Jones interaction, the blue one. So if I had a very simple fluid like liquid argon, okay, or xenon of real atoms, right, they would interact through some sort of a potential that looks like this. Now, there's an old theory of simple fluids called the Weeks-Chandler-Anderson theory that tells us the structure of simple fluids is largely dictated by that big wall there. You know, in the Lennard-Jones potential is a 1 over r to the 12th power that blows up when you get on top of each other. The Weeks-Chandler-Anderson theory says, it's remarkable actually, all simple fluids, argon, xenon, anything that's a simple atom, even to some degree methane because the hydrogens are like little bumps, okay? It has a coordination of 12 neighbors. So if I sit on one of those particles, I will uniquely get 12 around me on average. And it's all dictated by that hard wall there. It's packing. Okay, it has nothing to do with any interaction. It has nothing to do with this out here. It's just how they pack at temperature. All right. What does water do? So this I love this example just because it's kind of interesting. Remember, what I tried to do here is I said I'm going to force water to act like liquid argon, but water's not going to want to act like liquid argon. Water wants to act like water. So it's going to pop up into something going on here. So what does it do? Water, of course, is tetrahedrally coordinated, right? Water has 4 neighbors. It's kind of an expanded liquid. It's this weird liquid, you know, as you make it cold, it expands. It puts that bump, that jig in the potential right in the middle of where this famous theory for simple fluids tells us everything is defined by right in there. Water says, you can't do that. I'm not going to let you do that to me when you try to coarse grain me. Okay, when you coarse grain me, that is going to make me expanded and be a tetrahedral fluid. This is the radial distribution function. There are different things in here. Don't worry about all the curves. Just look at the blue one. That first peak, okay, if you integrate the area under that peak, you get 4 neighbors. That first peak is right there. And then we get this very deep minimum here. You have to be a little bit careful with this is a radial coordinate, right? So as I sit on one water, I'm going out radial, but I have all this space, you know, radial coordinate is volume is expanding. So even though this is very deep, there's entropy as I get larger, and that leads to this additional structure out here. So water illustrates to us what's interesting about this coarse graining at a very fundamental level. We start really believing these interactions as being effective interactions of real physics mapped into these simpler objects. All right? So let me take this a step further. If these are now free energy of interaction, these coarse-grained interactions, so this effective potential, I told you, is what we call a potential of mean force. It's a free energy. That means it has an energetic, and entropic part, right? You remember you learned the free energy is, Helmholtz free energy is energy minus Ts. Gibbs free energy is enthalpy minus Ts. So we're using Helmholtz free energy here because it's constant volume. That means that I should be able to split these coarse-grained interactions into an energy term and an entropy term. And that should give us some insight. That actually maybe be very interesting. So I'll show you the example. This is something that you do in your computer lab. A little bit less complicated than water, although in principle there's no reason why it is. Methanol is an amphiphile, right? It's got OH here, and it's got hydrophilic group there, or hydrophobic group there. I'm going to do that same crazy thing. I'm going to take methanol and map it into a fluid of single particles with this coarse graining. And I just want to, the reason I'm doing this is I want to show you how intensely subtle and interesting this coarse graining business is, all right? When I do that, the effective interaction at the coarse grain level between these single particles is given by this black line here. Okay, this is the pair interaction. Remember, I still have a fluid of these things. I'm only just showing one of them, but they interact through this black curve. It looks a little bit similar to water. Remember, water had that blip right there, but also went very deep. Okay, so that's methanol. Now, to make this a little more interesting, I'm showing also the decomposition of that black curve into the energy part and the entropy part. The energy part, and I show them as delta E because they go to 0 out at long distance from each other. The energy part is attractive the whole way. Okay, it's attractive. It then goes up, has a barrier, and then gets very attractive. And of course, eventually it will go up. We didn't carry this up, but eventually you can't crush these things on top of each other. Now, why is that? Well, what this coarse-grained fluid has to represent when the particles are getting close to each other is what really happens physically when two methanols get close to each other. All right. Now, what really happens when two methanols get close together is that they start attracting each other at the energy level. Right. There's OH will interact with another OH, and also flipping things over, the methanols will interact with each other. So that's this attraction. Interestingly enough, at some point there becomes a clash, right? It actually goes up. There's a bit of a little barrier there. So presumably that's some sort of a clash where orientationally the methanols are hindered a little bit. And that's in this interaction. What is even more interesting, in my opinion, is the entropy part. So the entropy part, we're plotting it as minus T delta S, because that's how it appears, of course, in the function. So what we're looking at is the minus T. The delta is relative to out here. The entropy term is purely repulsive. So the entropy term goes, and it's repulsive, and there's a blip here, pretty well aligned with where there's a blip here, and then of course it goes up. Now, why is that? Think about it physically, and think about how you might use this to understand what's going on at a deeper level. I'm mapping methanol into these single particles. I'm coarse-graining them. As they get close to each other in the fluid, all right, at first, you know, they have lots of freedom, but as they start getting close to each other, they start hindering the freedom of each other because they start having clashes. So they can't just freely rotate to them. That manifests as the entropy, right, going down when you hinder motion. Delta, that's going the wrong direction, right? Second law of thermodynamics, you're not supposed to do that, right? So minus that is going up. And so I get a net repulsion. Okay, so this is a really interesting example of what's really behind coarse-grained modeling, all right? I mean, at a rigorous level. There's entropy, there's energy, they're in these coarse-grained interactions, and it's very difficult to understand them without separating them out. And I'll give you one example, then I'm going to have to move forward pretty quickly here. This is, again, going back to lipids, this is kind of an ugly picture, I'm sorry. This is a coarse-grained lipid, okay? A little bit different than the one I showed before. And I'm just showing you this because many coarse-grained models, they started with modeling lipid bilayers, and many of them from different programs model the coarse-grained interactions with a Lennard-Jones potential, okay? Just because that kind of makes sense. These carbon chains, I coarse-grain them, I fit it by some sort of a simple Lennard-Jones potential, which looks like this black curve here, all right? This is what the actual interaction between, I think it's these groups with groups like them in a full lipid bilayer. I'm just showing one lipid here. So the actual free energy surface, or free energy function, is this black curve. It looks very much like a Lennard-Jones potential. But when I separate it into the energetic part and the entropic part, you can see those are rather different. They're fighting each other. The black is a compromise. Again, we can kind of understand this. The energy term, it goes out. There's a little bit of a hump here, and then it's pretty attractive at close range. Why? When these chains get close to each other in a lipid bilayer, they start kind of lining up and having a lot of interactions with each other. Just think of the CH groups interacting across, so they get lots of attraction. The entropy terms, though, really don't like that, right? When they start getting close to each other, it starts hindering the freedom of those chains. And so the entropy starts climbing. It becomes repulsive way before the energy does. The entropy likes being out here where they have more freedom, the chains to move around. It's more entropy. Entropy is good, right? So the net compromise is the black line, but it's made up of two pieces. So this shows you, I think, the subtlety of this kind of interaction at the coarse grain level and why it's so important to be able to try to get this accurately. Yeah?
I probably missed something. Why is entropy effective in the long range?
Yeah. So first of all, I probably should have shown more than one lipid. You're in a lipid bilayer, right? And there's a lot of freedom of the tails in the lipid bilayer. As they get close to each other, there are clashes which hinder their motion, so the entropy goes down. When they're a bit further apart, they have more freedom, so entropy goes up.
I mean, I understand
This here.
the repulsive part.
No, I'm talking about
Entropy is, again, delta T.
minus T delta S. Yeah, so if entropy goes up from the reference point, then it's going to be attracted because there's a minus sign. You see, so delta S would, and I'm sorry, the delta is relative to the out here where it's 0.
Well, why is it unfavorable at really long distances?
Oh, out here? These are, you have to be careful. These are pair interactions. So the real lipid bilayer is the sum over all these possible interactions. This is just between two of them. So interpreting these out here at long distances is not that meaningful. Okay, it's a decomposition of all the pair interactions. I'm just showing two of them. It's not as if that is representing a real lipid bilayer has been turned into a gas. Okay, it's just how one pair turns off. But there are many other pairs that are not. So yeah. Okay, I'm going to have to move forward because I want to get to. These slides are be in there in this talk when you get it. And these are a little more technical. Looks like. One thing I want to talk about, though, and this sets the stage a bit for tomorrow. One notion about coarse graining is that actually you don't need to retain the solvent. So I talked about these ideas of doing this coarse graining. When you, and this is a very, very important step for large systems that when we want to simulate, very large protein systems, because the solvent, there's a huge amount of it. So in three-dimensional space, if you have a lot of proteins floating around, it turns out that you spend a ton of time just calculating solvent-solvent interactions. So one notion of coarse graining is that we would like to just get rid of the solvent. But there's nothing in this methodology I told you about that stops you from doing that. In other words, it all could be used to coarse grain systems and just eliminate solvent. Why does that work? Well, the effect of the solvent gets folded in to this effective interaction. So the fact that this solvent is now gone, it pops up into these interactions. I'll show you quickly just an example of that. I don't have time to show this. This is a lipid bilayer where, in fact, the solvent's gone. All right. So there is no solvent. It's kind of a crazy idea, but because a lipid bilayer is driven by amphiphilic assembly, right? The tails want to be together, the heads want to be in water, but there's no water anymore. But the idea, though, is that the effect of the water, the driving force, can be folded into the coarse-grained model. This is all coarse-grained lipid. There's a lot of them. It's about 10 million atoms, effectively, but there's no solvent. So you can definitely do this kind of thing. Let's see. I want to advance. I'm not going to have time to talk about the issue of dynamics. We may come back to this someday. And what I want to finish with is a little bit about to sort of set the stage for the rest of the talks today and part of the morning. Where we want to head with all this work, and really the whole purpose of this center here, is to push these concepts really upward in scale. All right. This is a picture I've used many, many times of an actin filament. So eukaryotic cells have all these actin filaments in the cytoskeleton. These are long, long filaments of protein. They're made up of this monomer, which is the actin monomer going into these filaments. And this is just a small piece of a filament. I think there's 13 monomers there. And that already, if you just try to do a brute force attack on that, It's about 700,000 atom simulation. So with full water solvation and just 13 of these in a periodic simulation box, you get about 700,000 atoms. So what we really want to do is we want to coarse grain it. And this introduces a sort of a new concept, which I'll call ultra coarse graining. You see, what's different about this problem is that whole regions of the protein have been turned into one of these coarse grain objects. This is a good illustration. I'll explain this diagram in a minute. This is an actin monomer, okay? And you can see that we have different coarse grained beads. They represent, many amino acids. They may represent somewhere from 10 to 20 amino acids, as opposed to 4 or 5 atoms. So there's a question of how you even do these mappings in the first place, let alone the issue of how they interact with each other. All right, I spent a lot of time in this talk describing how we model and discover what these coarse grain interactions are. At this level of coarse graining, there's a question of how you actually and where you put these coarse grain beads and what amino acids they represent. So what we're working on in this center is an attempt to sort of tie everything together that I was telling you about, and I'll sort of describe this a little bit. The way we have to work when we're at this level is, it's not quite as pure as what I told you about before. We cannot just start from the ground up and just like magic pop out a coarse grain model and we're off and running, okay? What we have to do is first sort of build coarse grain models from experimental data, everything we have in terms of structural data. Sometimes it's using electron density. These are some electron density maps of actin filaments. And the way this basically works is that we then develop sort of what I call a first pass coarse grain model. And you're going to have a chance to play around with one of those in your computer lab for HIV, the HIV virus capsid. This is for actin. And what we're working on then is, after we've developed sort of a first pass coarse grain model, to use that coarse grain model to explore the space, to explore the complexity of the problem, the key interactions. And then this sort of ties into the talks you're going to hear after the break. So the Martin McCullough following me is going to talk about how we do these coarse grain mappings. Then after the break, we'll talk a little bit about then taking these coarse grain models to enhance our sampling, to calculate at a more detailed level the atomistic interactions that go into the coarse grain model. So tying in one way or the other to the first part of the lecture I gave today. So sort of actually, as opposed to starting from the bottom up, starting now more from the top, working back down to the bottom, refining our coarse grain model, and then you can keep looping, okay? You can keep going and going as you get more and more experimental data. You can keep refining and getting a much better coarse grain model. And the goal, of course, is to be able to study something as ambitious as this and really push up to the scale of cellular biology. So I'm going to show you one example. I don't think I have time to show both, just of how these sort of, this is kind of a fun application, and you're going to have a chance to play around with this, I think, tomorrow afternoon. In HIV, there is HIV, the key sort of bad player is a very long polyprotein called the GAG protein, and it has different domains to it. And this long protein, the top part of it, called the matrix domain, targets the cell membrane. This is an artist's rendition of a cell membrane here. This part of the HIV virus targets a special lipid called PIP2 in the cell membrane and starts nucleating on the cell membrane. And then because this part of the protein is sort of broad, this is, again, an artist's rendition. You can see sort of this shoulder here. It starts bending the cell membrane. And it creates this what called a viral bud. These are all these proteins in there. There's RNA in there, and this thing is budded. And then what happens is some other proteins are recruited and it cuts this neck, and this thing comes off. And this is a viral particle. This is called the immature virus. So what happens then inside this virus is the HIV protease, which is an enzyme, comes and it cuts this thing where these arrows are shown. I think the sequence is that it cuts here first, then here, then some of these other places. This core piece, the CA part, is called the capsid domain. That forms dimers. I'll show you those in a minute. And those dimers encapsulate in some sort of a very interesting lattice the RNA of the virus. It's called the virus capsid inside of this viral particle. These are some images. This is an immature particle here. This is a mature particle. I think I have a better image in a minute. This one. These are images of an infectious HIV particle that goes and targets other cells. This is this encapsulation by proteins of the RNA of the virus. And it's made up in its shell of these dimerized proteins. They have a really interesting structure. 1/2 of them is that. That's 1/2 of this dimer. After this cleavage, there's a very strong interaction between two of these right in here. And it forms that, and that goes into the shell. Interestingly enough, the structure of that shell looks like a carbon fullerene. Have you all seen buckyballs and all? So these aren't carbons. These are just the lattice structure of the proteins that are packing in this lattice. And it has some high curvature regions where they have pentamers. So as opposed to hexameric structures, there's pentamers here to allow this curvature. And so this is what is believed to be occurring in this. This is a very coarse-grained model. It's just an artist's model of the proteins. There's a dimer, and there's a dimer, and there's a dimer, and there's a dimer. And they form this helical structure with a kind of a chiral spin to it to make this lattice. And so what I want to finish my lecture with today is just to show you how some of this coarse-grained modeling is making a difference in this kind of thing. It's certainly not the final word, but I'll just show you this. And if all goes well, you'll be able to run some of these simulations on Tuesday and do crazy stuff. Maybe you'll discover something new. So what we have created are coarse-grained models of these dimers. You can see the coarse-grained beads there. All right. And there's some subtleties in how you do this modeling. It ties very much into mining the structural database and modeling the attractive versus the repulsive interactions. But at the end of it all, we've been able to create a coarse-grained model of the virus capsid. This is this thing at temperature. You can see the pentamers. We color them red. It's structurally stable. If you look closely, you'll see the hexamers. The C termini, where they're dimerized, are kind of a layer beneath in the gray, and the hexamers are there in the green. And we see these different structures. I'll come back to a minute. This is a hexameric structure. This is the pentameric structure. This is the all-atom representation, not the coarse grain. Just to make a long story short, though, what I wanted to tell you about is going back to this idea of how we want to push forward this modeling. This is at the level of making a first-pass coarse-grained model. And I told you, once we've done that and we create a good coarse-grained model, we start looking for key interactions. And let me show you where that has led us with this particular problem. When we scan the possible interactions that hold that virus capsid together, what these graphs show, so there's a two-dimensional plot of, this is the interaction between the N-terminal domains of each of these. This is the C-terminal with the N-terminal. Along this is the C-terminal with the N-terminal. This is the N-terminal with the N-terminal. What this shows is the logarithm of the root mean squared deviation from that perfect virus structure. So in other words, we can do a huge number of simulations, like I just showed you, that movie. We can vary these parameters and understand the stability. Okay, so blue means it's very stable. It's like a rock. Okay, we've glued this stuff, and of course that's stupid. I mean, nothing's like a rock. That's just that we pushed the parameter space too far out. What is interesting is then this region where it becomes unstable versus stable. And what we discovered from this is that the most stable structure in that lattice, the most important one, is not that hexamer, not the pentamer, but this thing, which is what we call a trimer of dimers. Right, so there is one of these dimer proteins there's another, and there's another. Okay, and it's a trimeric packing. And in the HIV lattice, actually, they're all over the place. Okay, you tend to think about these hexamers and these pentamers, but there's this trimer, 1, 2, 3. You see a triangle there? So sometimes there's structure underneath what you really think is important. It turns out that in this lattice, the most robust piece is actually that. And we've gone on to show that, in fact, if you explore the early assembly phases of the HIV lattice, so what this is projecting down on a two-dimensional simulation, you'll see the trimers all over the place, okay? And the trimers, so there's a flying trimer right there, they're all over the place, but every now and then they nucleate the hexamers. And once they nucleate the hexamer lattice starts growing. These are, by the way, 4 periodic replicas, so the fact that there are 4 things that look alike shouldn't make you think there's anything special. We just have to fill the screen here, okay? But what you find is that the stable structure are these trimers, a little bit hard to see there. I'll show you another movie in a minute, flying all over the place. They're forming and breaking apart all the time, but they really are the key component for the nucleation. And every now and then they come together to start nucleating that hexamer lattice that I showed you about. And this I'm going to accelerate this simulation artificially by every time one of these trimers comes together, I'm going to glue it together. That's unphysical, okay? So don't quote me that that's the right thing to do, but it just accelerates the movie. Every time they come together, I force them to stay together, and you'll see how important they are for growing this lattice. Okay. So you see the trimers all over the place now, right? So then they start, and I don't know if you're going to be able to play with this two-dimensional simulation, but you start growing a hexamer lattice all over the place. Like you see this really nice, there's hexamer, hexamer, hexamer. They keep adding to the edges. So it appears with this coarse-grained modeling, we've actually been able to discover the critical nucleating structure of the HIV formation in the virus. And it just so happens a company has some kinetic data that supports this, and they've been working on an inhibitor that binds in here, and it seems to really inhibit the HIV virus. They didn't know how or why it works, but now we think we know why. So I'm going to have to skip the rest of this. I want to leave just a couple minutes for, whoops, sorry. I'm going to skip all of this. I just want to mention one other thing because you're going to play around with this. Another very important problem that we worked on is membrane remodeling. In endocytosis, that means neural synapse behavior of sending off some sort of something and taking it into a cell. There's a very important set of proteins called bar domain proteins that bend membranes. And the reason I need to mention this is in one of your computer labs, you're going to coarse grain this thing, okay? And these bar domain proteins are really a very hot topic. You can, in vitro, you put them in vessels with liposomes, and they do all kinds of crazy stuff to liposomes, remodel them into these tubulated structures, very interesting behavior. So that's one of your, you'll be doing these coarse-grained models. And so I'll just skip that. I have to say one thing, though. This is this outcome of the simulation with these coarse-grained models. You can see how it's very fast. I'm sorry that it tears up this liposome. What we found, though, is something at very low concentration, something really unique, that these proteins like to form strings. So this is a lipid bubble that they're on top of the surface of, and they like to form strings. But once they form a string, they encircle part of the lipid and start pooching it out. And we think that this is the first discovery of their role in the early phases of endocytosis, which is bending the membrane and creating some sort of a structure that, that other proteins can bind to.